So absorption of water and ions, let's see what we've covered so far. We looked at the small intestine, the fact that it's got villi, as well as crypts of Lieberkuhn, the villi for absorption, the crypts of Lieberkuhn for secretion, with the stem/progenitor cells, rectangular shaped, for development, and the Paneth cells at the bottom for the immune function. So, a quick comparison to the large intestine cells, of the fact that they don't have any villi, they have more goblet cells, and they don't have crypts of Lieberkuhn. Instead, they've got intestinal glands in the mucus layer with surface epithelium, colonic enterocytes, and they also have stem cells for development. Looked at the fact that there's 3 layers, plicae circularis, which are the folds of Kirk-Ring, which are the macroscopic folds, the villi, macrovilli, to increase the surface area. And then had a look at the overall digestive tract absorptions and secretions, the fact that nothing is absorbed in the mouth or the esophagus or the stomach. Most of the absorption happens in the small intestine and then partly in the large intestine, leaving you with 0.1 litres at the end. Looked at how water is absorbed and the fact that it's absorbed via tight junctions as a result of the absorption of sodium and chloride. Ions can be absorbed through 2 routes, essentially transcellular through the cell, which is active, or paracellular, which is between the cell, which is passive. Sodium is absorbed throughout the intestines, but mainly in the jejunum. And there's 4 routes: sodium glucose, sodium amino acid cotransport, sodium hydrogen exchanger, parallel electroneutral sodium hydrogen exchanger and chloride bicarbonate exchanger, as well as epithelial ENaC sodium channels in the colon. Sodium is absorbed along with glucose and amino acids, using energy from the importance of this ATPase channel, sodium potassium ATPase. And the cotransport happens through this cotransporter SGLT1. Sodium is also transported in via sodium-hydrogen exchanger mechanism. So the hydrogen goes out, the sodium comes in, and this is stimulated by bicarbonate, because you want to neutralize the bicarbonate by secreting the acid. And those 2 mechanisms were in the fed state, whereas in the fasted state, the sodium is absorbed in the ileum and colon by parallel sodium-hydrogen and chloride bicarbonate exchange. As shown here, the bicarbonate and hydrogen coming from the carbonic anhydrous reaction. And the fact that this absorption is inhibited by CAMP from enterotoxins, as well as angiotensin, which stimulates it. Looks at the fact that enterotoxins, such as cholera, they cause diarrhea by elevating levels of CAMP, which inhibits sodium chloride absorption from the parallel sodium-hydrogen exchange and chloride bicarbonate exchange. Finally, sodium is absorbed in the colon via sodium channels, these ENaC channels in the colon, and it's highly efficient because of this gradient created by the sodium-potassium ATPase. Aldosterone causes or stimulates sodium absorption by causing more channels to be inserted, more to be made, and more to remain open. So overall, aldosterone stimulates absorption. Chloride ions are absorbed via 3 routes, so passive, parallel sodium-hydrogen and chloride bicarbonate exchange, just like sodium, and chloride bicarbonate exchange. It's passively absorbed throughout the intestines, and it's driven by energy from the sodium glucose and ENaC channels, causing passive chloride absorption. It's absorbed in the ileum and the colon via parallel sodium-hydrogen and chloride bicarbonate exchange, and this is in the fasted state. And again, this is inhibited by CAMP from enterotoxins, Werner-Morrison syndrome, which is a vipoma, a vasoactive intestinal peptide-secreting tumour, CGMPN, and calcium. Calcium is also absorbed without the parallel sodium-hydrogen exchange via just the chloride bicarbonate exchanger, and the proton, instead of being secreted into the lumen, instead goes back across the basolateral membrane. Looked at congenital chloroduria, which is autosomal recessive. We have an absence of this chloride bicarbonate exchanger, so you're going to secrete less bicarbonate, absorb less chloride, so you're going to have high stool chloride and increased plasma bicarbonate, which makes you alkalytic, which you can treat using proton pump inhibitors through this alkaline tide method. Potassium is absorbed in the ileum and the jejunum via 3 mechanisms: paracellular, passive, as well as solvent drag when it's pulled in by water. Whereas it's secreted from the large intestine via potassium channels stimulated by aldosterone. So the potassium ATPase, sodium potassium ATPase channel causes a gradient, causes potassium to be pumped in, which then leaves into the lumen via the potassium ion channels. And it's stimulated by aldosterone. So aldosterone stimulates the absorption of sodium chloride, whereas it stimulates the secretion of potassium. Looked at how you can control absorption and secretion using absorptagogues and secretagogues. The absorptagogues being angiotensin and aldosterone, causing the increased sodium chloride absorption, with angiotensin acting on the sodium hydrogen exchanger, whereas aldosterone stimulating the ENaC sodium ion channels. And then looked at the 4 kinds of secretagogues, bacterial enterotoxin, hormones, immune cell products and laxatives, raising the levels of CAMP, inhibiting absorption, promoting secretion, ultimately resulting in diarrhea. Cholera-toxin, for example, is an enterotoxin, causes diarrhea by increasing CAMP, therefore increasing chloride and potassium secretion and reducing sodium chloride absorption, resulting in diarrhea, which you can deal with a rehydration solution containing sodium chloride powder. Hormones such as VIP from vipomas, from Werner-Morrison syndrome, can also cause, are also secretagogues. And immune cell response as a result of the deglutination of mast cells into histamine and prostaglandin also causes diarrhea, which you can treat by using somatostatin analog to inhibit the ECL cells from producing histamine, or you could use loperamide, which inhibits the myenteric plexus to decrease the motility, so there's more time for water to be absorbed. Laxatives are the last kind of secretagogue, and we looked at bulk laxatives such as methylcellulose, osmotic laxatives such as lactulose, which are fermented in the lumen, and stimulant laxatives such as senna, which stimulate the enteric nervous system to increase peristalsis. Finally, we looked at calcium absorption and the fact that it can be passive, which is not controlled by vitamin D receptor, or it can be active, which is controlled by vitamin D receptor, which and what happens is the calcium enters the enterocyte, binds to calbindin as a buffer, very important, and then it's pumped across the basolateral membrane to be absorbed translaterally, transcellularly even. And if you have a vitamin D deficiency, it will cause rickets, and you'll have a lack of calcium absorption, which you can treat with sunlight and fish oils. And the vitamin D deficiency will mean that you won't activate the vitamin D receptor to allow the calcium absorption.